Chronic cerebrovascular diseases constitute an ever-increasing problem in the aging population. Hypertension, hyperlipidemia, diabetes and atherosclerosis cause latent blood supply disruption leading to cerebrovascular diseases which can vary widely in terms of symptoms and clinical manifestation. Stenosis of blood vessels results in a decrease in the levels of glucose and oxygen supplied to tissues. This leads to a significant impairment of neurometabolism. A reduction in the levels of glucose and oxygen activates the ischemic cascade, a series of biochemical reactions triggered by inadequate blood supply to cerebral tissue. The release of cytochrome C activates apoptotic pathways and increases oxidative stress, whilst activation of the NMDA receptor may activate pathways leading to lipid, protein and DNA damage. Ultimately, these processes result in irreversible neuronal injury and cell death. Currently available treatment options for chronic cerebrovascular diseases typically only deliver symptomatic relief. However, a number of neuroprotective alternatives have been tested for their ability to improve cognitive and neurological outcomes following cerebral ischemia. Actovegin is an evidence-based neuroprotective therapeutic approach for patients with acute and chronic cerebrovascular diseases. Actovegin possesses neuroprotective properties and a highly favorable safety profile. Besides Actovegin being widely used in neurological practice, its efficacy for the treatment of cognitive disorders of various origins is specifically supported by evidence from 6 clinical trials in various types of dementia. Actovegin contains more than 200 biologically active physiological components such as oligopeptides, oligosaccharides, vitamins, minerals, and eicosanoids. Being a multi-component drug, Actovegin is believed to have a multifactorial mode of action. In previous experimental studies carried out by Parade and co-authors, Actovegin was found to have an effect similar to insulin, enhancing glucose uptake by the cell. Actovegin was also shown to stimulate oxygen transportation into the mitochondria and to stimulate cellular respiration, increasing cell survival under hypoxic conditions as proven by de Groot and co-authors. An in vitro experiment conducted recently at the University of Tübingen in Germany has suggested that Actovegin has specific neuroprotective effects in neuronal cultures. Using a rat hippocampal neuron model, researchers investigated the effects of Actovegin on dendrite length, oxidative stress, apoptosis, neuron survival, and the number of synaptic connections. Increased levels of reactive oxygen species are well known for their detrimental effects on proteins, DNA, and intracellular structural components. In the Tübingen University investigation, reactive oxygen species were stimulated using tertiary butyl hydroperoxide, TBHP. 2 days after cell cultures were treated with tertiary butyl hydroperoxide, a considerable burden of oxidative stress was achieved as measured by the levels of reactive oxygen species. Actovegin was found to reduce the level of reactive oxygen species significantly in a dose-dependent manner to a level lower than the control cultures without exposure to TBHP. Apoptosis, programmed cell death, is considered to be another important outcome as well as an indicator of degeneration in the CNS, including chronic ischemia. The activation of the protease caspase 3 is an intermediate step in the apoptotic signaling cascade, leading to cell death, and is a suitable marker for monitoring the level of apoptosis presence. In the Tübingen University study, caspase 3 activation was induced using a synthetic amyloid beta peptide 2535, analogous to that known from Alzheimer's disease. The results revealed that levels of activated caspase 3 were reduced in the presence of Actovegin. Finally, neuron and synaptic numbers are the most important factors impacting on neuronal plasticity and neurological function. The researchers from Tübingen found that neuron survival and the number of synaptic connections increased dose-dependently in the presence of Actovegin. There was no difference in dendrite length and synaptic numbers per cell between the groups. The efficacy of Actovegin has also been tested extensively in clinical studies. Cognitive performance and other endpoints related to quality of life improved in 6 clinical trials testing Actovegin in dementia of various types. In an aging population, it is important to treat cerebrovascular diseases as early as possible and tackle the underlying causes and not just the symptoms. An upcoming randomized double-blind clinical trial will investigate the efficacy and tolerability of Actovegin treatment in 500 patients who have recently experienced a stroke. The study will aim to demonstrate the efficacy of Actovegin in the prevention of cognitive decline, as well as prevention of dementia, and demonstrate its disease-modifying mode of action. With dose-dependent effects such as reduced reactive oxidative species levels, reduced apoptosis markers, improved neuronal survival, and increased synaptic numbers, this study corroborates the findings of previous clinical trials, which have shown that Actovegin improves cognitive performance and quality of life in patients with dementia. The new clinical trial in post-stroke cognitive impairment will provide further evidence of Actovegin's benefits in the patients who recently experienced a stroke. Actovegin brings back vital energy to the nerve cells.